Hi kids, it's Mrs. Fravel again. How are you? I hope you're fine. I hope you're good. If you're working at home online due to all of this wondrousness we call the COVID-19 pandemic, this video is for Monday, February 22nd. I apologize for not having very many videos in this unit. It's been kind of crazy. So I just want to make sure that everybody is on the same information level in terms of what we've talked about so far in this unit. We're talking about Duchenne muscular dystrophy in particular, and what causes disorders like that in general. So far last week, boy howdy, we've covered a lot. We've covered so far how muscles work, what the protein dystrophin does in muscles. Tell me what does dystrophin do? Go ahead and say it out loud. Nobody's listening. It's fine. Yes, dystrophin is a protein that links the actin cytoskeleton inside of a muscle cell, also called a myocyte. It links that actin to that muscle cell's sarcolemma or cell membrane, so that when the whole muscle contracts, that actin and the membrane, they move together rather than what happened. Oh, sorry. In people who have muscular dystrophy, that dystrophin anchor or hook or rope or fishing line or kite string, or however you wrote an analogy about it, those were really good, by the way. That dystrophin hook does not link the sarcolemma to the actin. So when the whole muscle contracts, what happens on a cellular level is that those cells get ripped apart. The membrane actually tears. And normally in healthy cells, when muscle tears happen, the muscle gets to work fixing that and rebuilding it and building more muscle cells so that they get stronger. Unfortunately, due to the result of the tearing from the dystrophin being not functional, calcium rushes into the muscle cell and causes protease to destroy that muscle cell's healthy proteins. And also creatine kinase rushes out of the muscle cell, leaving the muscle cell with no energy to contract more or rebuild or heal itself. So people with muscular dystrophy end up having a lot of scar tissue, and they don't build muscle tissue after it has been torn or after doing exercise. So it's a rough one, right? We've talked about some of the ethics behind should people with muscular dystrophy do physical therapy. If you have not done that assignment, go do that. Okay. And then we talked about how are proteins made? Like, how is a protein like dystrophin made? And what can happen, or what might cause a protein to not work? And we determined that through protein synthesis during transcription and translation, which is the process inside of a cell that builds a protein. If the amino acid sequence in a protein is incorrect for some reason, the protein is not going to work. The protein won't be the correct shape, it won't be the correct size, and it isn't going to work. So you guys asked some really good questions in your discussion check-in on what do you think's going on, or what else do we need to investigate? And most of you came up with what we're going to talk about today. Why is the dystrophin not being built properly? What's wrong with the gene? Okay, so that's what we're going to investigate today. Woohoo! Okay, so let's get going. First of all, you had a starter that you should have already done that was labeled in Canvas starter for today, lesson 3, part 2. Why don't people with DMD make dystrophin? And you should have read a paragraph about dystrophin and the DMD gene. So let's just make sure you got where we should be. So I'm making sense today. I'm good. It's fine. Okay, so what did you take away from part 3's reading in the worksheets? And I apologize. I've just gone back through those worksheets. I'm relabeling it. It may not say part 3, but it will probably say DMD and the dystrophin gene. Okay, you'll figure it out. Don't worry. What did you get from the starter is what I'm saying. Still awesome. Okay, so what you should have seen was there is a gene in your DNA that codes for dystrophin, for that protein rope, hook, anchor, whatever, for dystrophin. And it is called the DMD gene, which starts to get confusing just word-wise, because if you don't have muscular dystrophy, if you don't have DMD, it means your DMD gene is functional. It's fine. And people who have muscular dystrophy, either DMD or BMD, there's a couple of different kinds, their DMD gene must have a problem. I mean, that's probably what you should have inferred, right? Duchenne muscular dystrophy, people with that disorder don't build dystrophin at all. And people with Becker muscular dystrophy, BMD, they build dystrophin that's just short. It's just not quite long enough to hook the sarcolemma and the actin together. So what should you have inferred from that? I just said it because I'm getting ahead of myself as usual. There must be something wrong with the DMD gene in people who have muscular dystrophy, right? So that's what we're going to investigate today, is the question that is behind this. What is a broken gene or a dysfunctional gene? Generally, we call that a mutation. And how does that happen? So we're going to investigate how mutations happen and what they are. Okay, so you should, if you haven't already, pause me and go open the note guide for this. It's actually another slide deck that I have left blanks in for you to fill in from this. So make sure you have that open. You can split your screen or fill it in below me if you want to work in the tiny screen. Okay, right back. Great. All right. What the heck is a mutation? So we usually think of mutations, thanks to Hollywood, we usually think of mutations as like these grotesque deformities or, superpowers, X-Men, am I right? But in actuality, mutations happen all the time. Like your body just made a mutation, just did it again. There's a mutation somewhere right now going on in your body because your DNA is replicating itself all the time and making mRNA all the time. And sometimes there's a mistake made, and all that mistake is a change in the base sequence, either on DNA or on our mRNA. And those bases, remember, the A's, C's, T's, and G's in DNA, and the A's, C, U, and G in RNA, that's the bases, right? And they match up complementarily, A to T, C to G in DNA, A to U, T to A, C to G, C to G, G to C in RNA. Remember that? Yes. Okay, good. Those, sometimes there's mistakes made. If you did, and you should have done the virtual lab on protein synthesis where it had you click the right nucleotide to match to build those base pairs. Did you make any mistakes? Like, did you click on the wrong one and then go, oh no, it's that one? Okay, so that happens all the time, for real. The DNA polymerase, which is and RNA polymerase, which are the enzymes that do the copying, they sometimes make mistakes. And your cells have like editors and double checkers, but they're not always caught. So a mutation is just when the wrong letter is put into a DNA or an RNA molecule when it's being built. Now, sometimes that is no big deal. It happens all the time. Sometimes, depending on when it happens, what kind of mistake it is, and what cell it happens in, that can cause a serious problem, okay? If it happens during mRNA transcription, the codon might be wrong. It might be read wrong in the ribosome, and it might call for the wrong amino acid. If that happens, if the wrong amino acid is in the sequence of a protein, what happens? Say it. Yeah, the protein doesn't work, right? The protein won't be the right shape, size, fold. It can't fit to do its job, okay? So Not all mutations are harmful, but some of them really are. They're catastrophic. Just even a small mistake can cause something as devastating as muscular dystrophy. There's all sorts of other disorders that it can cause, cystic fibrosis as well. And these are specific kinds of mutations, okay? There are different types. So it depends on what is going on with with the nucleotide sequence, okay? So types of mutations include a deletion. The polymerase just forgot one. It just missed one, just left one out. Or it put in an extra one. So deletion leaves out a base. Insertion puts in an extra one. What happens is what's called a frame shift, and that just shifts how the codons are going to be read, okay? So that means that the wrong amino acid might be called for by the ribosome, okay? So frame shifts, when they are harmless, actually drive evolution, which we'll get into when we talk about evolution. Frame shifts are responsible for slight variations in traits in humans. So if you, and other organisms, but we'll talk about people because you're a people. If you look around at the people around you right now, if you're by yourself, go find a person and look at them. Do they look exactly like you? Look at the little things, like the shape of their nose, the shape of their eyes, eye color, hair texture, height, skin color, freckles, all of those little things. Like, we're still people, we're still functioning humans, but we all look a little bit different, right? You might have your mom's nose, you might have your dad's eyes, it might be slightly different from your friends, your sisters might be opposite. But that is what causes humans to look different. Those frame shifts create different structures, just different variations in our structures, okay? So those are the harmless kind. The catastrophic kind are the ones that cause diseases. Okay, so there's other types of mutations. So deletion, insertion, point mutation means that there is a substitution. So one base is substituted for another base. So instead of linking a C to a G, it might link a T to a C. It happens. And or an A to a G, or an A to an A, or a T to a T, thymine dimers. So this one change, one mistake, these cause these three types of mutations: missense mutations, silent mutations, and nonsense mutations. So this can change the protein altogether, and it can or not. So it can change the amino acid. That's a missense mutation. It will make, instead of, say, serine, maybe threonine gets thrown in there. A silent mutation, everything's fine. It didn't cause the amino acid to change at all. Remember on the code chart, in fact, I think I have one. Hold please. So on the code chart, what do you notice about these amino acids? Is each amino acid called for by one codon? No. These amino acids have, it's called redundancy. They can be called for, leucine can be called for by 4 different codons. C-U-U, remember how to read it? C-U-U, leucine, C-U-C, C-U-A, or C-U-G. It's the C-U that's important. The third one doesn't matter. If a ribosome reads C-U in a codon, leucine, right? If it reads U-A, tyrosine, unless it's the third one, UAU and UAC, tyrosine. But if it hits the UAA, stop, don't make anything, right? Okay, so those mutations, can either change the amino acid or not. If they do change the amino acid, it can cause that malformed or shortened protein. And then a nonsense mutation, that's when, a stop codon's accidentally thrown in the middle. Okay. And that just makes the protein stop being translated in the middle. or not quite far enough, and it causes the protein to be too short. Okay? There is a video here just that kind of illustrates all of the garbage that just came out of my face. So make sure you watch it. I'll link it underneath this. Okay? Okay. Any questions? Shoot me an email if you have a question. Okay. Okay. All right. We're going to practice this. What I would like you to do is pause me and go transcribe this sequence. It's on your notes slide deck. So transcribe that sequence into an mRNA molecule. Pause me. Pause me. Go do that. Are you back? Okay. So you should have transcribed that into an mRNA sequence that is T to A to U, C to G, remember, and G's to C's. So your mRNA should have been AUG, UGG, AAC, CGC, UGC, UGA. Did I get all those right? Sometimes, you know, my brain. Anyway. Okay. So that's your sequence for mRNA. Now I would like you to take that mRNA, pretend that you're pulling it out into the cytoplasm from the nucleus, go translate it into a protein. Ready? Go. Pause me. Are you back? Okay. So this is, and I hope you figured out to use the code chart, right? So the amino acid sequence that you should have gotten is methionine. That's always our start amino acid, start code. Tryptophan, asparagine, arginine, cysteine, stop. So short protein, really short protein. But that should have that, whatever this gene codes for in the DNA, this protein comes out of it and goes about its merry way and does its job in the cell, right? Or in the body. Okay, so what happens if we have a mutation? So quickly again, pause me. Transcribe this DNA sequence and see if you can figure out the change. Okay, so on your notes, when you transcribe it into mRNA, highlight the change. Okay, pause me. Are you back? Okay, great. So here's what you should have gotten: AUG, UAG, AAC, CGC, UGC, UGA. Where is the change? Circle. If we're doing it on paper, you would be circling it. The kids in class are circling theirs. Where's the change between this DNA strand and this DNA strand and this mRNA and this mRNA? I should have done a little circle. Sorry. It is right here, right? This A right there should have been a G. Okay. So let's see if that's going to affect the protein at all. So go ahead and pause me again, take that mRNA, use that code chart on the next slide, and get your amino acid sequence for this mutated strand. Okay, pause me. Back. Great. What'd you get? Oh, that's not a protein at all, right? UAG is a stop code. So this one mutation, this one letter change, this one point mutation caused a nonsense mutation. So yes, there was an effect. It caused a nonsense mutation. Does this build a functional protein? Will that protein do what this protein's supposed to do? No, it's not going to at all, right? Again, proteins, these are there for a reason. They cause the protein to fold, bond, and do its job. It can't do that down here. Right? Okay. How many nucleotides were changed? Just one. One. That's it. And it caused all of that. Do you think there might be something going on that we can link this to DMD? You would be right. DMD is a point mutation. So you are going to be, there's your code chart. So your assignment today is going to be a virtual lab in which you are going to be building some proteins from some different DNA strands and looking at some mutation types and what they cause. Sorry, click. Okay, so make sure that you read all of the instructions. This lab is not super intuitive, so let's take a look at it together really quickly. Okay, so this is your home page on Canvas. So pause me, go find the home page and go to today's page. Pause me. Okay, you back? Great. So here is the assignment or the worksheet that goes with the lab. Always open it. You're going to save it in your biology folder on your Google Drive so you can access it quickly later, right? And you do that by clicking, there will be a little icon. There it is, this little folder icon. Put it in your bio folder. Okay, so protein synthesis simulation. So let's open this up. Again, you're going to need to be flipping back to answer questions. Let me see if I can get out of the way. Okay, so again, there's not a lot of instruction on here, but the most important thing is the first thing you have to do is push the setup button. That starts the experiment. And then hit the go stop button to watch it build. Okay, so essentially what you're going to do is once you have this open is first you're going to hit setup. Okay, it gives you a DNA sequence in this box. And look, it built that DNA sequence right there. That's that sequence. Okay, and it's showing you the genes. That's the, let me make sure I'm right. Maybe not. So then you're going to hit go stop, hit transcribe. There's your mRNA. Release. There's your mRNA strand going out into the nucleus. Translate. Oh, that was a fast ribosome. So there's your amino acid sequence and release. Okay, so go through that, answer the questions, and follow the instructions. Pretty simple. And then we'll have you run some mutations as well. Okay, so this gene that we just did is the gene that builds lactase. You'll also be running it for DMD later on in the worksheet. So again, make sure you read all of the instructions, answer all of the questions, do all of the things, and that's all for today. Have a great day. Be good, be kind, be smart, and be safe, and I hope to see you soon. Okay, bye.